Hello, my name is Tomas Lopes, and I am a 2022-2023 Choose Development Fellow. I'm part of the lab of Dr. Rahul Kanadia at the University of Connecticut, and I'm a rising senior. I began my research as a Choose Development Fellow, studying mouse limb development in the context of minor intron splicing, which is a primary focus of the Kanadia lab. What most people in our field ask us is, what's a minor intron? But what most people not familiar with the mechanism of gene expression ask first is, what is splicing? Quick overview of splicing. mRNA transcribed in the nucleus contains protein coding and non-coding segments, known as exons and introns respectively. There are two types of introns, removed from the transcripts by two different splicing machineries. The Kanadia lab focuses on minor introns, which constitute less than 0.5% of all introns. They have distinct characteristics, which means they are recognized by different spliceosome components. This minor spliceosome catalyzes the reactions required for the removal of these introns using small nuclear RNAs, or snRNAs, which recognize the unique minor intron sequences. The snRNAs are complexed with proteins and facilitate the recognition and removal of minor introns as well as the joining of adjacent exons, as detailed on the right-hand side of the diagram. But if there are so few minor introns, why are they important? The short answer is, all of your cells require them. Loss of U11, the snRNA that recognizes the 5' splice site, is embryonic lethal, meaning the genes that require the minor spliceosome are vital to development. Conditional knockout mouse that is U11 null in the developing limbs only allows us to study the significance of the minor spliceosome in one aspect of development. The list of minor intron-containing genes include important players in the cell cycle, and without them, limb progenitor cell numbers are reduced compared to the wild type. The cell cycle is slowed, and as a result, the limb is malformed. In addition, cell death increases, which further depletes the number of limb progenitor cells. To visualize the disruption of development in more detail, I performed immunofluorescence on 4 limb tissue sections for both the wild-type and U11 null mutant to mark SOX9, MyoD1, and neurofilament, which are markers of bone, muscle, and axons respectively. I imaged these sections and used a program called Reconstruct to create 3D renderings of SOX9, MyoD1, and neurofilament positive regions. These reconstructions highlight the severe defects of the mutant limb and showcase the interconnectivity of these systems during development, since loss of U11 occurs in the limb only, affecting the SOX9 positive chondrocytes that originate in the limb, but not the migratory muscle progenitors and not the axons extending into the region. I presented this work at the Society for Developmental Biology 82nd Annual Meeting. My current work has shifted to plant development. Alternative splicing is important to stress response and development in plants, an example of which is alternative splicing of a floral initiation gene, FLM, in response to temperature, controlling inflorescence development. It has been previously established that the minor spliceosome is required for proper development in plants, exemplified by the Arabidopsis U11/U12-65K protein knockdown, which exhibits vegetative and floral growth defects. However, its role in the stress response and development under stress has yet to be sufficiently explored. This is the gap in knowledge that my research aims to fill. RNA-seq data for Arabidopsis and corn show that minor introns are much more responsive to cold stress than other types of introns, suggesting that regulation of minor intron splicing may play a role in the cold response. This responsiveness refers to alternative splicing or retention of minor introns in mature mRNA. I hypothesized that the responsiveness of minor introns to cold shock could be due to differential expression of minor spliceosome components. If the introns are being retained at a significantly higher rate, I believed that the snRNA or protein components of the spliceosome would have lower expression. This deficiency in minor spliceosome components would therefore reduce minor intron splicing activity and result in the increase in minor intron retention. To test this, I decided to grow Arabidopsis in a hydroponic setup. There are some cold responsive genes that also respond to drought stress, and so to ensure only cold response is being observed, the ample water availability of a hydroponic system seemed like a logical solution. For the cold shock experiment, I simply moved the grow box into a cold room at 4 degrees Celsius, and then harvest 3-week-old plants after 1, 12, and 24 hours for RNA. Finally, I want to take some time to talk about how I chose development. Going into college, I knew I wanted to pursue research in something related to biology, and I had a passion for genetics and plants. I was introduced to developmental biology in my first year, joined the Kanadia lab. The field has only become more intriguing since. We take the ability of organisms to put themselves together for granted, but if you stop to think about it, development is just a ridiculously complex and organized chemical reaction that perpetuates itself in a predefined manner, consuming resources and responding to outside forces with the ultimate goal of further propagation through reproduction. Development is the basis for all life, and understanding the foundation of how organisms are generated from a single cell is an integral part of all biology, and technically all fields of science, as science itself is performed by organisms. I chose development not only because of its significance, but also because of the sense of wonder I feel when learning about it. And when I can spark that same feeling of wonder in others, I feel like I've achieved my goal.